So many people have been asking, hey Medicosis, when are you going to continue your endocrinology playlist? Let's do it right now. Today it's time to talk about cell signal transduction. Like, what's the difference between a cell surface receptor and an intracellular receptor? What are these doofuses doing here? This is my endocrinology playlist. Please watch these videos in order. Today is video number 7. Any company has a CEO followed by a general manager who has employees and independent contractors, who are the ones that are dependent on the manager employees, but the independent contractors are independent. Similarly, the CEO is your hypothalamus. General manager is the pituitary under the hypothalamus. And we have 3 employees that listen to the pituitary and 3 independent contractors that could not care less about the pituitary. If the pituitary wants to speak to the thyroid, the order is called TSH. If the pituitary wants to speak with the adrenal cortex, it's called ACTH. If the pituitary wants to have a word with the gonads, this is called FSH and LH. But how do you think the pituitary communicate with these? It doesn't. The hormones secreted by the employees are slower, but the hormones secreted by the independent contractors are faster. Let's say I'm running from a tiger. Boom, epinephrine. Oh, that's a very quick response. But let's say I want to grow and I need some thyroid hormone. Well, you're gonna wait some time. Let's say I need some cortisol. You'll have to wait until the morning. Charles Dickens wrote A Tale of Two Cities. Your body has a tale of 2 hormones, the lipid-soluble hormones and the water-soluble hormones. Lipid-soluble hormones are lipid, they are fat. Yet your blood is made of plasma, which is water. Fat cannot float in the water, therefore we have to carry it on a plasma protein. But once we reach the cell, since this is lipid, it can cross through the lipid bilayer cell membrane. It's phospholipid. So you go inside, you'll find your receptor intracellularly. Contrast that with the water-soluble hormones. Water-soluble hormones are water. They can float freely in the plasma. They do not need a plasma protein. Go to hell. Once they reach the cell, they cannot enter because they are water-soluble, but the cell membrane is lipid. Water cannot diffuse through the lipid. Therefore, you have to have a receptor ready on the outer surface of the cell, and this is called a cell surface receptor. How do lipid-soluble hormones cause action? Well, they keep diffusing until they reach the nucleus, and they tell the nucleus to do transcription, translation, etc. But how do water-soluble hormones do their action? Well, they cannot enter. Therefore, they need a messenger, like a second messenger system. Yeah, they need a second messenger. And who's gonna take the signal from the outside to the inside? This is the function of the G protein. Another analogy for the 2 types of hormones is the first engineer and the second engineer. The first engineer, in order for that engineer to turn on the light, the engineer has to walk and reach the house, enter into the house, and then turn on the lights. That's a lipid-soluble hormone. You have to wait until you diffuse, until you reach the nucleus. This is a very slow process. That's why the 3 employees had slow actions. However, an independent contractor can just flip a switch. You turn it on without even having to go to the house. This is the story of a water-soluble hormone. Just from outside, turn on a switch and the nucleus will do what you please. But in order to achieve this, you need some electrical transduction. Yeah, but here we call it signal transduction. And that's the story of the G proteins and the second messengers. Lipid-soluble keep diffusing until you reach the nucleus. The nucleus has genes. We call this genomic action. But the water-soluble hormones cannot enter. This is non-genomic action. Where should I put the receptor? Well, it depends. If you're waiting for a water-soluble hormone, put the receptor outside, cell surface receptor. But if your hormone is lipid-soluble, it's going to diffuse through the membrane, and you can put the receptor inside the cell, whether in the cytoplasm or the nucleus. The ligand is the hormone. This could be water-soluble or lipid-soluble. What's the receptor? The receptor is a protein on which the ligand will bind. The ligand alone is not active. The receptor alone is not active. But when they hug and kiss each other, ligand-receptor complex, they are active. Active to do what? All kinds of functions. You can open a channel, activate an enzyme, increase calcium intracellularly to cause contraction, to squeeze a gland and secrete hormones. You can do anything. The ligand alone is not active, the receptor alone is not active. That's why if I have a pathology can hit me here, such as type 1 diabetes, or it can hit me here in the receptor, such as type 2 diabetes. Similarly, in diabetes insipidus, you have type 1, which is central diabetes insipidus, and you have type 2, which is nephrogenic diabetes insipidus. Do you lack the ligand, or do you have a defective receptor? Type 1 versus type 2. Easy peasy. These three are employees. Their secretions are slow. Why? Because they are lipid-soluble hormones. If they are lipid-soluble, they cannot float freely in the plasma, and that's why someone has to carry them. We have thyroid-binding globulin, cortisol-binding globulin, estrogen-binding globulin, testosterone-binding globulin, collectively known as sex hormone-binding globulins. Contrast that with those independent contractors, water-soluble, fast action. They can float freely in the plasma. Have you ever heard of a parathyroid hormone-binding globulin? No, because it does not exist. Neither does an epinephrine-binding globulin or an insulin-binding globulin. These are urban myths. Let's compare between lipid-soluble versus water-soluble hormones. Lipid-soluble, they listen and obey the anterior pituitary. Polar hormones, pooh-hoo, I don't listen to anybody. I work for myself, I'm independent. Examples of lipid-soluble hormones include thyroid hormones like T3 and T4, suprarenal cortex such as aldosterone, cortisol, adrenal androgens, and sex gonads secreting estrogen, progesterone, testosterone. The water-soluble hormones are the product of the parathyroid gland, PTH, adrenal medulla, epinephrine and norepinephrine, and dopamine, pancreas, insulin, glucagon, somatostatin. What are the nature of these hormones? Alright, chemically speaking, they are steroids, fat, or thyroxine. How about these? Chemically speaking, they are water-soluble components, usually proteins or peptides. Lipid-solubles have to be carried on plasma proteins, but the water-soluble float freely in the blood. Lipid-solubles can diffuse through the membrane by simple diffusion, therefore the receptor is internal in the cytoplasm or the nucleus. However, water-soluble, they cannot enter, the receptor has to be external, aka cell surface receptor. But once they flip a switch, it's flipped. It's very quick. The corticosteroids, the sex hormones, the thyroxine, all of these are lipid-soluble. The proteins, peptides, pituitary products, pancreatic products, parathyroid products are water-soluble. Look at this lovely amino acid tyrosine. Tyrosine is just magic. Why? Because it can give you thyroxine, which is lipid-soluble, or it can give you catecholamines, which are water-soluble. Same freaking amino acid. Lipid-soluble hormones in the plasma are bound to plasma proteins. The bound portion is physiologically inactive. Since you are bound to a protein, you will not get lost in the urine. Conversely, polar or water-soluble hormones are freely floating. The portion that's freely floating is physiologically active, rapidly acting. Since they are small in size and they are not bound to plasma proteins, they are lost in the urine. Let's talk about the lipid-soluble. Okay, I'm here on a plasma protein. Most of us are on plasma protein. Only less than 10% is floating free in the blood. Almost non-existent. Once I reach the cell, I will leave the plasma protein and then jump onto the cell membrane, diffuse until I reach the nucleus. What are you doing in the nucleus? I'm tampering with hormone receptor elements, stimulating the DNA. DNA becomes another DNA called replication, or DNA becomes RNA, transcription, RNA becomes protein, translation. This is a genomic action. But what about the water-soluble hormones? They cannot enter, they have to stay outside. Where's the receptor? The receptor's outside. Cell surface receptor. How can I join the receptor on the outside with the enzyme on the inside? You need a middleman. What's the name of the middleman? The G protein. The G protein's job is to connect the receptor on the outside with the enzyme on the inside. What is the name of these enzymes and micromolecules? Second messengers. Then who's the first messenger then? This hormone is the first messenger. What is signal transduction? It's the process of converting the extracellular signal into intracellular signal and then action. What is signal amplification? Amplification means exacerbation, stimulation, resonance. I'm increasing the number of signaling molecules, such as these, at every step of the signaling cascade. Please remember that all of the receptors in your body are made of proteins. What are the second messengers? Intracellular, inside the cell, enzymes and small molecules. Let's review the IP3-DAG system. Take, for example, Mr. Angiotensin II. What does angiotensin II does? Well, it tries to constrict vessels. How do you constrict vessels? I go to the alpha-1 receptor and tell the alpha-1 receptor, hey, big boy, do your job. Alpha-1 receptor is now stimulated by the ligand. Who's the ligand? Angiotensin II. Who's the receptor? Alpha-1 receptor. What kind of receptor is this? GQ-coupled receptor. Here is a mnemonic for you. There is a magazine called GQ Magazine, and it has muscular dudes. Why are they muscular? Because they are contracting their muscles. How do you contract? Calcium. GQ, calcium. It's the Q and the C. Speaking of C, here is phospholipase C. Phospholipase C destroys phospholipids. Let's break down the phospholipid. Where do you get the phospholipid from? From the lipid bilayer, the cell membrane. Oh yeah, that makes sense. It has to be the cell membrane because these are cell surface receptors. They are not intracellular. They are on the outer surface of the cell. So that makes sense. Break down that fat. Get me that PIP2. And then I will break it down into IP3 and DAG. This is the third letter in the alphabet, and this is number 3. What does that do? I increase calcium, and which will activate protein kinase C. What will calcium do? Calcium is the hero of contraction. Of what kind of muscles? Smooth muscles. Like the one in my bronchi? Yeah. Like the ones in my blood vessel? Yeah. That's why angiotensin II constricts your blood vessel, raises your total peripheral resistance, and raises your blood pressure. See, medicine makes so much sense once you understand what the flip you're talking about. What does freaking oxytocin do? Oxytocin contracts the uterus. The uterus is a smooth muscle because you have no voluntary control over it. So smooth muscle, you need calcium, you need phospholipase C, you need to be GQ coupled. It makes perfect sense. A third example, V1 receptor of ADH. ADH is going to bind the V1 receptor. Awesome. What's the result? The result is vasoconstriction. How did you do this? I did this by stimulating alpha-1, GQ coupled, phospholipase C, IP3, calcium contraction of smooth muscles in my blood vessels. Bingo. How does gastrin work? It squeezes the glands in my stomach in order to secrete. How do you squeeze glands? By contracting the myoacini, very small, smooth muscles surrounding the glands. When you squeeze the gland, it secretes all kinds of secretions. That was the first story, the story of IP3-DAG system. Now, to the second story, the cyclic AMP story. Let's go. We start by a GS-coupled receptor. Let's take an example to make it easy. Beta-1 stimulation, which is supposed to increase heart rate and contractility. How does that happen? Imagine that I'm running from a tiger. Okay, what do you like to do? I would like to increase heart rate and contractility. Amazing. I'd like to dilate blood vessels going to skeletal muscles and going to the heart to get more blood supply. Awesome. I would like to raise my blood pressure and retain the fluid. I don't want to go to the bathroom right now. So renin angiotensin II, which will raise your blood pressure and give you aldosterone to retain salt and water. So let's do this, people. Beta-1 is being stimulated. It's GS coupled. Why S? Because it stimulates. Stimulates what? Cyclic AMP formation. How did that happen? I stimulate adenylate cyclase. Adenylate cyclase converts ATP into cyclic AMP, giving you protein kinase A activation. And this will lead to what? Increased contractility of heart muscles. Not smooth, cardiac muscles. What will you do to the smooth? Contraction? Heck no. Contraction was the calcium story. I'll do the opposite. I will relax them. Bronchodilation, vasodilation. Even the uterus will relax, called tocolysis. If mommy is contracting her uterus prematurely, we do not want to deliver the baby right now. Hey mommy, wait. Hey uterus, stop contracting. What should I do? Give mommy a tocolytic agent, which is a beta agonist. Tocolytic decreases uterine contractions. Sympathetic stimulation also increases renin and increases aqueous humor secretion. So GS-coupled receptor, everything here is A. Adenylate cyclase, converting ATP into cyclic AMP, activate protein kinase A, increase contractility of the heart, bronchodilation, vasodilation, uterine muscle relaxation. And now to the beautiful chart. Hormone signal transduction pathway. We have intracellular receptor, for the lipid hormones. We have receptors on the surface for the water-soluble hormones. We have others, such as non-receptor tyrosine kinase. And as you know, the second messenger systems are cyclic AMP, cyclic GMP, IP3, and DAG. We talked about cyclic AMP, we talked about IP3 and DAG. This was the first story. The second story, we'll talk about this soon. Just remember, cyclic GMP goes hand in hand with nitric oxide. We have intracellular receptor, cell surface receptors, non-receptor tyrosine kinase or NRTK, the JAK-STAT pathway, and second messengers. The cell surface receptor are subdivided into three categories: G protein, ligand-gated ion channel, enzyme-linked receptors. The enzyme-linked receptors are divided into two categories: receptor tyrosine kinase, not non-receptor tyrosine kinase, actually receptor tyrosine kinase, and this is the MAPK pathway, and receptor proteins. Remember the sulfur-containing amino acids, serine and threonine. Now it gets ridiculous. Who uses the intracellular hormone lipid-soluble hormones, who can diffuse through the lipid membrane via simple diffusion? Your steroid hormones include aldosterone, that's the adrenal cortex, cortisol, adrenal cortex, adrenal androgens, zona glomerulosa, zona fasciculata, zona reticularis. Also, we have testosterone, progesterone, and estrogens from the gonads. You have thyroid hormones, T3 and T4, as well as vitamin D. Next, cell surface receptor. G protein coupled include acetylcholine, catecholamines, parasympathetic and sympathetic. Insulin is here. Who's anti-insulin? Glucagon. There is a protein that secretes secretin, and there is a protein in the serum, serotonin. It's also in the brain. How about ligand-gated ion channel? This is GABA-A and acetylcholine. If you remember your pharmacology, how did benzodiazepines work? They worked by stimulating the GABA-A receptor, which opened chloride channels, ligand-gated channels. Third, enzyme-linked receptor, receptor tyrosine kinase, and receptor proteins. Receptor tyrosine kinase is the most important thing in the entire stinking slide. Never ever forget that insulin and growth hormone are receptor tyrosine kinase. Anything that's pro-growth and pro-anabolism is here. Insulin wants your fat to grow. It wants your skeletal muscles to grow. IGF-1 wants your bones to grow. Fibroblast growth factor wants your fibroblasts to grow. Platelet-derived growth factor lets the platelet grow. How about the receptor protein, serine and threonine? This is TGF-beta, which is a cytokine. Non-receptor tyrosine kinase, think of a baby. What does the baby want? The baby wants to breastfeed, prolactin, and grow. Growth hormone. Growth hormone is here. Growth factors are here. What else does the baby want? The baby wants to build bone marrow. Okay, let's build red blood cells. EPO. Let's build platelets. TPO. Let's build granulocytes. Granulocyte colony stimulating factor. What are the granulocytes? Remember BEN. B-E-N. Basophils, eosinophils, and neutrophils. Alright, how about the non-granulocytes such as my lymphocytes? For this, you will need interleukins, such as interleukin-2, interleukin-6, and interferons. Second messenger system, we mentioned them briefly. We will elaborate in the next video. But let's talk about this cyclic GMP nitric oxide. What are you trying to do? Vasodilation. Do you remember this story? Yeah, how does hydroxy nitroprusside nitrate, sildenafil, tadalafil, vardenafil work? They basically work by the nitric oxide cyclic GMP system. What are you trying to achieve? I'm trying to activate protein kinase G because this is the cyclic GMP. Remember, the previous story, the story of cyclic AMP, we had adenylate cyclase, ATP, cyclic AMP, protein kinase A. Yep, A was the story. But here is the story of the G. Guanylate cyclase, GTP, cyclic GMP, protein kinase G. What are you trying to do? I'm trying to relax smooth muscles. Where in blood vessels you get vasodilation, and in erectile tissue you get erection. And that's why it's contraindicated to take nitrates and sildenafil. What's the problem? Both of them dilate vessels. What's the problem? When you dilate vessels too much, you get severe hypotension, which can kill you. And if the severe hypotension did not kill you, the reflex tachycardia will. And that's a common exam question. A patient was taking a medication for erectile dysfunction, but he also had a heart disease and was taking nitroglycerins. If you take both together, you can die. What are the intracellular receptors? These are the lipids. Steroid hormones, think of your adrenal cortex and your gonads. Thyroid hormones and vitamin D. We'll continue this chart in the next video. Pause and review. If you like this video, I have an endocrine pharmacology course on my website, medicosisperfectsnatus.com. We talk about all the insulins, all the oral hypoglycemic drugs, etc. To learn about anti-hyperlipidemics, anti-arrhythmics, antihypertensives, etc., check out my cardiac pharmacology course, also at medicosisperfectsnatus.com. And for a limited number of students, I have a 40% discount towards anything on my website. Just use promo code KIDNEY. Thank you for watching. Please subscribe, hit the bell, and click on the join button. You can support me here or here. Go to my website to download my courses. Thanks for watching. Be safe, stay happy, study hard. This is Medicosis Perfects Natus, where medicine makes perfect sense.